Don't go past Model 2. Like, don't start the questions about Model 2, but you can do all the questions about Model 1. Okay. I'm actually going to stop you in just a second, but. Okay, even if you're not totally done with the questions about Model 1, I'm going to pause you there because I want to make sure we have time to talk through all of these pieces today. So we're going to talk a little bit about this diagram just to make sure that you kind of understand what's going on. Like I said, this packet's your notes. So if you want to go back and finish off the questions after we talk about it or change or add to anything, that's fine. If you feel like you understand it enough, you don't have to. This is your packet to keep. So this type of operon, this is called an inducible operon. What does that word inducible mean? Able to be started, able to be turned on. So you might want to even write that up here. And on has an N at the end there. And the really important thing to notice about that, if it's able to be turned on, how is this operon usually? It's usually off, okay? And that's an important thing to realize. Inducible operons tend to be used for clusters of genes that are only necessary to the bacteria in certain situations. And the most common example that you see of an inducible operon is the lac operon. It talks about that one in your note packet. It's got that little blurb about it. But this type of operon is frequently found for like digesting substances like lactose. So lactose is a sugar, and when it's around, bacteria can digest it and use it for energy. And it takes enzymes to break down that molecule. But if there's no lactose around, why would you bother wasting energy in your little tiny cell making enzymes that don't have a function, right? So the point of operons really is to help bacteria be as efficient with their energy and their resources as they can, so they don't make things they don't need. So most of the time, this type of operon is off. So this is kind of the normal situation in this top picture. When this is off. So you can see here the regulatory gene, that's the one that in that other picture was labeled with the letter I. The regulatory gene produces the repressor protein. And in this case, the repressor protein is active when it's made, which means it's in the correct shape to go and bind to the operator. So that active protein goes up here, binds to the operator, and prevents transcription. Because most of the time the cell doesn't need these genes to be functioning. They don't need these proteins, perhaps the enzymes to digest lactose. If there's no lactose, you don't need to make those. But if that substance becomes available, if we're talking about the lac operon, if lactose is suddenly available in the environment, then you want these genes to turn on and make those enzymes so the cells can break it down and use it. So the lactose itself, or a molecule that's usually found with lactose, usually acts as the inducer molecule. So here we could kind of go through and narrate this and say, number one, if lactose, if that's our example, is present in the environment, then we'll go down here, number two. It binds to the repressor and makes it inactive by changing its shape. Remember, you have to think back to what you know about protein structure here. The repressor is a protein. And remember, protein shape determines its ability to function, and its shape is determined by the chemistry of all the amino acids that make it up. And those amino acids have all kinds of chemical interactions going on that determine how it folds into that shape. And so if something else bonds to it, like if a molecule of lactose bonds to that protein, the chemistry of that lactose molecule is going to alter the interactions and the bonds within that protein and change its shape. In this case, it changes its shape in such a way to make it inactive, so it can't bind to the operator. It doesn't fit up there anymore. So there's nothing in the way. So now RNA polymerase can transcribe and translate. So transcription occurs and translation happens. And you make these enzymes, these proteins, X, Y, and Z. And just to kind of finish this story and bring this full circle, the enzymes you're making, remember if we're talking about the lac operon, we're making enzymes to break down lactose. So these genes will turn on, they'll start making these enzymes, and these enzymes are going to go and start breaking down this lactose, which means over time the amount of lactose within that cell is going to decrease. And eventually there will be so little lactose that there's no lactose to bind to the repressor protein anymore. That'll be broken down and digested. And then when there's no lactose, then there's nothing bound here anymore. That repressor protein will go back to its active shape. It'll bond to the DNA and it'll say, okay, no more lactose. We don't need to make these enzymes anymore. And turn it back off. Questions about this one? Okay. If you turn in your packet, the next page has a similar but not exactly the same picture labeled Model 2. This one is another type of operon called a repressible operon. I'm just going to give you a couple of minutes to talk about this one. You can start answering some of the questions, or you can just study this diagram and talk through it together. And explain what's going on and how this one is similar and different from the other one. Again, you can answer all the Model 2 questions. Don't go to Model 3 quite yet. Okay, no, you may not be totally done with that one. Again, we're going to keep going to make sure that you understand what these are showing, and then you can go back and kind of finish it up later. So this one's a different kind of operon. Instead of an inducible one, this is a repressible operon. So what does that mean? Able to be turned off. So how is it usually? On. So this type of operon, a repressible operon, is used usually for the production of substances the cell needs. The example that's in your packet, kind of the most common example, is the tryptophan operon. Tryptophan is an amino acid, and just like we need all 20 amino acids to make our proteins, bacteria do as well. And so most bacteria have the ability to synthesize, to make their own tryptophan, and it requires these enzymes, protein QRS. Those are the enzymes needed to build tryptophan. And the cell needs to have a constant supply of tryptophan so it can make proteins. So usually this operon is turned on, which means the regulatory gene is producing a repressor protein, but this repressor protein is inactive when it's made. And so it can't bind to the operator. So usually RNA polymerase is free to do its thing. It transcribes and translates and makes those enzymes so the cell can continuously produce the tryptophan that it needs. But if that bacteria suddenly finds itself in an environment where there is tryptophan just floating around it, why would it bother making its own if there's a bunch of tryptophan sitting right in its environment? So it has the ability to turn these genes off when there's plenty of tryptophan available, or when it's made enough that it doesn't need any more for a while. So we can kind of do this again. If the substance, maybe tryptophan, is available in the environment, so that would be number one. It binds to the repressor protein and activates it. So the repressor protein turns the genes off. It blocks transcription so that those genes are no longer being made and the cell doesn't produce tryptophan when it already has plenty of tryptophan. And just like the other one, this one kind of naturally undoes itself. Like, as long as there's tryptophan in the environment, these genes will be off. But then if this tryptophan all gets used up, or if it like moves out to an environment where there's not so much tryptophan, then suddenly this co-repressor molecule is not there. The protein goes back to being an inactive form, and those genes turn back on, saying, okay, we ran out of tryptophan, we got to start making some more again. Questions about this one? So both Model 1 and Model 2, both inducible and repressible operons, are examples of what's called negative control. It's called negative control, and I think you need to add this in your notes somewhere. It's called negative control because when the protein bonds to the DNA, it stops, it has a negative effect on transcription. Whether that's how it normally is or not, when that repressor molecule bonds to the DNA, it always turns transcription off. That's why it's negative control. As with everything in biology, in reality, this is more complex than one or the other, or just this or that. There's always more to it. So there are some genes that have multiple layers of control. And so there is also a type of positive control that we're just going to talk about briefly. The reason cells need multiple control mechanisms is because real life is more complicated than just you have something or you don't. Sometimes, like with the lactose thing, sometimes the cells have lactose, but they also have glucose, which is actually a better energy source. So they'd rather break down the glucose. But then after the glucose is gone, then they want to turn on the lac operon so they can digest the lactose. So cells have a lot more levels of control than just one or the other. So the third model in your packet, we're just going to talk about real briefly here. I know we're running short on time, but we'll talk about the third one. It's okay. So both of these are negative control because when the repressor binds, it slows down transcription. Positive control, which is the third model, is when a molecule binds that makes transcription happen more or faster. So here you can see that the regulatory gene makes something called an activator protein. So now it's not making a repressor, it's making an activator. Here's where this gets kind of fun. This one right here is an inactive activator. Isn't that a fun phrase? It's an activator protein, but it's not doing its job yet. Without an activator protein, operons that have an element of positive control don't work as well. The RNA polymerase either can't bind to the promoter without an activator to help it, or it doesn't bind very well or very often. So without an activator, there will be little or no transcription. basically, RNA polymerase just doesn't really know where to go, and it can't grab on really well without the help of this activator. For some operons that have this element of positive control as part of their regulation. When this molecule gets turned on, so again, it takes a signaling molecule to activate the activator. Fun, I know. The activator then goes and binds to the promoter, right at the beginning there. And it helps to increase transcription or make it go faster. So that's positive control. When we do the lab next week with bacterial transformation, it will have some of these control mechanisms built in as part of this. Before you totally stop listening to me, there is a homework assignment posted in Classroom today. It's a Google form with some questions about operons. I think I set the due date for Monday night, but I wanted you to have a chance to think through this again because I know there's kind of a lot to sort through here, okay? So you can do it this weekend or you can do it Monday night. Have a great weekend, folks. Yeah.
When you're gonna start the homework, or if it will be like the matching biology thing?
I'm gonna look at it this weekend and decide what I'm gonna do for that. I may have you doing personal progress checks from College Board instead because I like to start reviewing for the exam. So we will be doing something, but I'm gonna figure out over the weekend what I'm gonna do for.
I was just wondering if there was one due like Tuesday or something.
No, there won't be one due Tuesday. This Operon homework is kind of gonna go in that homework category. So this is kind of the homework you have for now, and I'll let you know probably by Monday what the plan will be for the rest of that.
Okay, sounds good. Have a great weekend.
You too. Thank you.